Hello, I'm Sean Eakins, CEO and founder of Collaborations Pharmaceuticals. Thank you for joining us today for our webinar, Exploring Drug Repurposing for Alzheimer's Disease. In a few moments, we'll start the presentation, but I would just like to give you a little bit of an outline today in preparation for what's to come. As you'll see, there are some tools on the webinar software that you can use to ask questions. There's a little icon at the top of the screen, and there's also a chat bar on the left-hand side. And you can put your questions in there at any time, and we'll read them at the end. I'm going to provide a little bit of an introduction to Collaborations Pharmaceuticals, and then Patricia Vigneault is going to talk about the work that she's done on Alzheimer's drug discovery. And then at the end of that, I'll give you a little bit more of an insight into the kinds of services we offer at Collaborations Pharma, and then we can have the Q&A session. And that should give you plenty of time to put your questions together. All right, well, Collaborations Pharmaceuticals is a small company that I founded in 2015. We currently have about 10 employees, and we're located in Raleigh, North Carolina. And we have 3 laboratories here. One of them is BSL-2 graded, so we can do wet lab work. And we're based in a facility on the NC State incubator. We're currently a company that doesn't have any angel or VC funding, but we have been funded by grants through the NIH and DoD to date. And this has allowed us to build up a pretty sizable molecule pipeline, as well as capabilities for providing services around data curation and machine learning, which I'm going to give you a little bit more of an outline on today. This obviously makes us a pretty stable partner, as we've been around for a few years now. We've been able to file a number of patents around the molecules that we've identified, and also new molecule IP. And we've also been able to obtain 8 orphan drug designations on rare and neglected diseases with the FDA, as well as 2 rare disease designations. So what do we do at Collaborations Pharmaceuticals? Well, I like to say that we use our artificial intelligence alongside real drug discovery intelligence, namely the 24 plus years of experience that I have working in the industry. And we use this to develop clinical candidates for rare, neglected, as well as unmet therapeutic needs. We are essentially a drug discovery company, but we also provide contract services to other companies. And primarily these have been in the area of using our data curation as well as AI machine learning technologies to help these companies deliver results. We've primarily focused on early drug discovery and hit to lead candidate optimization. And we tend to work on challenging ADME-tox problems. We essentially provide the little diagram on the right-hand side, which is the steps where we curate data, build models, generate predictions with those models, and then perform experiments to validate the models, and ultimately feed the data back into the training set. We've been able to put together quite a large number of machine learning models in the order of thousands of drug discovery and ADME-tox properties. We've been able to put these together as little packages of models, as well as create proprietary tools, which allow us to propose new analogs of molecules and also assess their synthetic feasibility. Computational drug repurposing is a specialty of our company, and we have many publications in this area. Some of the collections of models that we've been able to curate include the following software: Assay Central, which is our core technology for data curation, as well as building and validating models; Megatox, which essentially pulls together all of the toxicology and molecular property models; Megatrans, which is a subset of human drug transporter models, important for understanding efflux as well as uptake of molecules; and finally, Megapredict, which is a collection of thousands of models focused around chemical data sets, which would allow us to use them for repurposing of new molecules for new novel targets. Our technologies and approach so far has enabled us to build a pretty robust pipeline, and we do have a focus on a number of neurological diseases, including Alzheimer's disease, Batten disease, CLN1, as well as sialidase. And these are all in our current pipeline at different stages. And as you can see, we've been able to employ machine learning in most of our projects, either to find the hit compounds or to optimize the molecules that we've identified. So without further ado, I'd like to introduce Patricia Vigneault, and she obtained her PhD in genetics and molecular biology at the University of North Carolina, Chapel Hill. And she first came to our attention as an impact student and worked with us as an intern in 2019. Then we hired her as a postdoc, and during that time she worked on the project she's going to present today. And then we recently hired her as a staff scientist at Collaborations Pharmaceuticals. And she's currently working on a DITRA-funded project around understanding the metabolism of pesticides and nerve agents. So without further ado, I'm going to pass it over to you, Patricia.
Thank you, Sean. Good afternoon, everyone, and thank you for joining us today. As Sean said, my name is Patricia Vigneault, and I'm going to tell you about what we've been working on for Alzheimer's disease drug discovery. It is no secret that Alzheimer's disease is a major public health concern. It is the most common cause of dementia, and it is currently the 6th leading cause of death in the United States. It affects 1 in 10 people over the age of 65, representing 5.8 million Americans and an estimated 35 million people globally. These numbers are expected to grow proportionate to increases in population and life expectancy. There is no cure for Alzheimer's disease, but there are a handful of medications approved for symptomatic treatment. In 2016, the nearly $3 billion market for Alzheimer's drugs was divided almost equally between acetylcholinesterase inhibitors and the NMDA receptor antagonist memantine. Formulation of memantine and donepezil was approved in 2014 for moderate to severe Alzheimer's disease, but no new active ingredients have been brought to market since 2003. That is not for lack of trying. Since that time, more than 200 therapies have been investigated for Alzheimer's disease and have either failed or been abandoned. To cut down on the time and cost to bring new therapies to phase 3 trials, many researchers have chosen to investigate drug repurposing as an avenue for finding new treatment for Alzheimer's disease. In drug repurposing, drugs that have been previously investigated or approved for one indication and reevaluated for treatment of another disease, either through direct mechanisms or through off-target effects. In a review from August this year, Rosen et al. Showed that 39% of drugs in clinical trials for Alzheimer's disease are repurposed and include such disparate drugs as blood thinners and antidepressants. There are 2 approaches for finding repurposed candidates for disease: finding drugs that address the phenotype or symptoms of the disease, and finding drugs that target a known mechanism or pathway of the disease. In our work, we use machine learning to help with both these approaches and to predict which drugs may have activity for a given disease. We mine public datasets for compounds that have been evaluated against a given disease or target, assign molecular fingerprints to molecules that do not interact with the target, create models that describe structure-activity relationships based on those fingerprints, and then use those models to predict the interaction of untested molecules with the target. We can then take these prediction scores generated by our models to decide which compounds to investigate in vitro. Collaborations Pharmaceuticals, we perform the data curation, modeling and prediction, chemical purchasing, and test our predictions in vitro, either in-house or with external commercial companies. We can then take the results that we've generated and feed them back into the machine learning models to strengthen the predictive power of the models. We have used our proprietary software Assay Central to create a suite of machine learning models to score molecules against several popular targets in Alzheimer's disease. Today, I will tell you about the progress we have made this past year on 2 targets, GSK3β and acetylcholinesterase. GSK3β is a protein kinase that is a drug target of interest in diabetes, cancer, asthma, arthritis, and bipolar disorder. It is also a very attractive target in Alzheimer's disease. It has been found at high levels in the brains of people who died of the disease. It has been implicated in the improper processing and clearance of amyloid precursor protein. Studies have shown that inhibition of GSK3β results in less neuronal death in mouse models of the disease. And most interesting of all, GSK3β is the kinase responsible for the majority of tau phosphorylation, which leads to the formation of neurofibrillary tangles. Currently, there are dozens of commercially available GSK3β inhibitors for laboratory use, but none of these are suitable for human use. Early attempts to make GSK3β inhibitors for therapies were abandoned due to adverse drug effects. To date, only 2 GSK3β inhibitors have made it to clinical trials. Deglizib, which was clinically safe but was halted after phase 3 trials showed no clinical benefit in Alzheimer's disease, and lithium, which has been investigated for psychiatric symptoms associated with Alzheimer's disease and is currently under investigation for preventing disease progression in people with mild cognitive impairment. One of the advantages of drug repurposing is that molecules with a safe dosing regimen are the starting point. To this end, we have used our machine learning model for GSK3β inhibition to perform a virtual screen and score the Superdrug 2 library for active compounds. This library is composed of over 4,000 molecules that have been investigated or approved for pharmaceutical use in the US or other countries. From this screen, we picked 5 compounds to test in vitro, which resulted in the identification of a sub-micromolar inhibitor of GSK3β. The models created by Assay Central assign both a prediction score and applicability to each molecule that is queried, based on their individual molecular fingerprints. Prediction score is a measure of how likely it is that the molecule is active against the target. The higher the prediction score, the more likely that the molecule will be active. The applicability is a measure of how well represented the molecule is in the model, and therefore can be seen as a measure of the confidence of the prediction score. The higher the applicability, the more of the molecule exists in the training set of the model, and the greater the likelihood that the prediction score is meaningful. From our screen, we chose 5 molecules with high prediction scores that were not antibiotics or chemotherapeutic agents. Shown center is a visual representation of how they scored in the model, with a color-coded box to the right of the molecule. Green represents a better fit. All of these compounds had a prediction scores greater than 0.5 and relatively high applicability. Our testing showed that the compound with the greatest prediction score and applicability, riboxystorin, performed to the best in our in vitro assay, inhibiting GSK3β activity 96.5% at 100 micromolar. 2 other compounds, dantrolene and nifedipine, showed more modest inhibition at the same concentration, while nefepan and darafenacin failed to inhibit. We performed additional testing to determine the IC50 of riboxystorin against GSK3β, shown on the left, as well as sent the compound out for evaluation from a third-party contract research company, shown center. What we discovered was that riboxystorin is a submicromolar inhibitor of GSK3β with IC50s at 39.4 and 155.1 nanomolar respectively. This molecule scored less impressively against our model for the other GSK3 isoform, GSK3-alpha, and this prediction of lower affinity was borne out by in vitro IC50 evaluation of 695.9 nanomolar. Riboxystorin is not an approved drug. It is a clinical stage compound developed by Eli Lilly as a protein kinase C inhibitor selective for the beta isoform. It was previously investigated as a potential treatment for pathologies related to diabetes, including retinopathy, neuropathy, nephropathy, and macular edema. It is currently under investigation for improving cardiac function in patients with heart failure. Because it has been through several rounds of clinical trials, its safety as a potential drug has already been established, even for long-term indications such as diabetes. In one particular study, it was proven safe in diabetes patients for 4 years at 32 milligrams per day. As it happens, we found that the GSK3β activity was previously described in a patent for the use of chemicals to modulate GSK3 signaling for treatment of bipolar disorder and other brain disorders. However, since this application was not published in a scientific journal and was not present in any of our publicly sourced datasets, riboxastorin was absent from our GSK3β machine learning model and was therefore a genuine success of the model. To date, no one has published any investigations of riboxastorin in the context of Alzheimer's disease, nor reported any animal work to the National Institute on Aging. Since in vitro work is preliminary, additional testing in cells and in animal models is required to determine if riboxastorin could be a future drug candidate for Alzheimer's disease. The second example of drug repurposing that I'm going to talk about is on another popular Alzheimer target, acetylcholinesterase. Acetylcholinesterase is one of two Alzheimer's targets for which there are approved drugs, so finding new acetylcholinesterase inhibitors seems like it should be a low priority. However, the low upfront investment of drug repurposing offers us the ability to search for other potential therapeutics that could be less expensive for the patient or have fewer side effects. We used our machine learning model of acetylcholinesterase inhibition to score the microsource spectrum screening library. This in-house library has 2,560 compounds that are drugs, natural compounds, or other molecules from the microsource discovered libraries. However, when our model predictions showed nothing outstanding, we switched tactics and performed an in vitro screen. We first screened the entire library for inhibition of acetylcholinesterase and found 3 previously uncharacterized acetylcholinesterase inhibitors. We chose the most potent molecule, tilirone, to investigate further with the human enzyme. We discovered that tilirone had submicromolar activity, with an IC50 at 73.3 nanomolar for human acetylcholinesterase, though it was not nearly as potent as the most common acetylcholinesterase inhibitor, donepezil, at 3.3 nanomolar in this assay. This activity was validated by another contract research organization at 56 nanomolar. In a check for specificity, tilirone had no activity against the closely related enzyme, vitrocholinesterase. Crystal structure of human acetylcholinesterase with donepezil has been reported, and we used molecular docking on the scaffold to compare how tilirone might inhibit the enzyme. We saw that tilirone is predicted to inhabit much of the same molecular space in the gorge of acetylcholinesterase as donepezil, and may experience similar pi interactions with tryptophan 286. Unlike donepezil, however, tilirone is predicted to have pi interactions with tyrosine 341 instead of tryptophan 86 at the bottom of the gorge. Whether this loss of interaction is responsible for the loss of potency of inhibition is unknown. Tilirone is of interest to us as it is an antiviral medication approved and marketed in several European and Central Asian countries, including Russia and Ukraine. Among its published antiviral activities include hepatitis B and C, herpes simplex virus, and influenza. Studies within our company have shown further activity against Ebola and Marburg, as well as chikungunya virus, MERS, Venezuelan equine encephalitis virus, and Zika virus. Now we have shown that tilirone also has potent activity against human acetylcholinesterase. In a follow-up panel, tilirone was tested against 44 targets important for drug toxicity and adverse events in humans, and showed that acetylcholinesterase was the only target affected. We also screened tilirone against a kinome panel of 485 kinases and saw no major inhibition at 1 micromolar. This suggests that tilirone is not broadly promiscuous. We have investigated several other targets for Alzheimer's disease with less success. We have tried to create meaningful models for glutaminyl cyclase, which is involved in amyloid beta post-translational modification, but the small data sets available for inhibitors means there is no real weight to our model, and a screening strategy would likely be best for this target. We also did some work with the third-party contractor to test a few predicted inhibitors for CDK5 and mTOR, both of which are involved in several pathogenic pathways in Alzheimer's disease. We did not discover anything novel. We are, however, not deterred, and there are certainly many more targets related to Alzheimer's with which we could take the same approaches described here. Hopefully, what I have shown you today is that through our drug repurposing efforts, both computationally and in vitro, we have been able to find several new inhibitors for Alzheimer's disease targets with submicromolar affinity. We did this in 1 year for less than $200,000. In that time, we have written 2 papers on our findings and submitted 1 patent. I think we have demonstrated that we could use Alzheimer's disease as a proof of concept for drug repurposing. If we can do it for this disease, there are certainly many more for which we could take the same approach. In the near future, we will be applying for additional grants so we can follow up on our findings and hopefully uncover some new candidates for repurposing. With that, I would like to thank current Collaborations Pharmaceuticals employees, Drs. Anna Poole-Rubio and Tom Lane, as well as former employees Eni Minerali and Daniel Foyle, for all the work they have done on these projects, and also Dr. Peter Madrid from SRI International, who collaborated with us on the acetylcholinesterase work. I would also like to acknowledge Biovia, Thermo Fisher Scientific, and Eurofins Pharma Discovery Services, whose products and services we used during these projects. And of course, I would like to acknowledge our generous sources of funding. Most of the funding for these 2 projects came from the National Institute of General Medical Sciences and DARPA. And with that, I would like to turn it back over to Sean.
Thank you very much, Patricia. Wonderful presentation. We're welcoming any questions that you might have, and in a couple of minutes, I'll be able to read those out and we'll be able to answer them. Just a reminder, you can use the button at the top of the screen or the attendee chat, and we'll go through them in a few moments. To give you a little bit of time to put those questions together, I just have a couple of slides that I'd like to share to give you a little bit of a summary of what we can do to potentially collaborate with you and some of our services. Obviously, as Patricia mentioned, we do a good amount of machine learning and data set curation. And the work that Patricia showed today was using Bayesian algorithms, but we obviously have other tools available, including deep learning, support vector machines, and random forests, as just a few examples. And we can work on data sets of various different sizes. So we've worked with relatively small data sets, less than 100 molecules, to upwards of hundreds of thousands of molecules in some of the libraries we've looked at. We can use our machine learning approaches both to prioritize compounds for synthesis or testing, and we can also rank many scores for predictions, as well as look at chemical diversity of molecules. In addition to the tools that we've developed in-house, we have other commercial tools for calculating molecular properties, and we have our own tools that we've developed, as I mentioned, for predicting synthetic feasibility as well as retrosynthetic analysis. So we're happy to accept any new challenges you may have. Please just get in touch with us. You can access some of our models if you'd like to give them a try at www.assaycentral.org. We have a number of models there, both for drug discovery targets, as well as off targets like P450s, and molecular properties. And obviously, we have a big interest in COVID drug discovery, so we have a number of antiviral models there that we're actively updating. If you'd like to find out more about our machine learning technologies, we've published a number of papers on the underlying core technologies, and in more recent papers, we've described a lot of applications to different areas. Why would a company or organization choose to work with CPI? Well, as I mentioned, we have complete freedom to operate. We're not under the limitations of angel or venture funders where we have to work on one project. So we have pretty much flexibility. And obviously, most of our work is grant funded, or we work on fee-for-service projects. And so that provides us with revenue so that we can continue to work. And certainly the IP that we create in a collaboration remains your IP. And our only concern really is delivering quality results for collaborators that we work with. We have a number of client use cases. We've worked with a number of multi-billion-dollar consumer product companies, primarily in the area of building and curating data sets for endocrine disruption. These have resulted in a number of models that we've published, as well as presentations at large international conferences. We've also worked with a mid-sized rare disease company, primarily around modeling the data sets and ADME-tox predictions, as well as delivering designs for new molecules for them. And we've also worked with a small animal model CRO. So I think now we can take a look at your questions. If you'd like to find out more, obviously you can contact us at the emails below. So let's have a look at the questions we have. I know that one of the questions we had was, will we be able to share our slide deck or recording? We'll be happy to make the recording available. We're not able at this time, because we've got unpublished data in there, to share the slide deck. One of the questions we have is, for repurposing, we could use the 505(b)(2) pathway. What are our options to examine safety in a clinical setting as soon as possible? Do the NIH offer any options? That's a good question. We, at least for Alzheimer's disease, we haven't explored those capabilities. I think probably one of the most straightforward approaches would be to collaborate with a clinician that specializes in this area. And that's definitely something we could do. We could approach clinicians with this work and see whether they could then obviously reach out to the companies that make the drugs to see if they would be interested in collaborating in doing that. Obviously, as the molecules that we've identified, such as riboxystorin, are made by quite large drug companies, I mean, that's certainly something we could do as well, is reach out to them. We're not just limited to options that the NIH may have. So, for example, we could certainly do a physician-sponsored clinical trial. Tilrone might be more difficult because that drug is approved outside of the US, but certainly riboxystorin, as that's already been through clinical trials in the US, that may be more of a straightforward type of collaboration for a clinical trial. Do you have any thoughts, Patricia, on other next steps for these molecules?
As far as Alzheimer's disease, I would think that next step before going to humans is obviously going to have to be go into some kind of animal study to look at it, because we do have the function in vitro of these molecules, but a lot of things have made it all the way through animal models in Alzheimer's disease, and then you get them into people and they haven't worked. So for these where we just have in vitro, I think the next step for us would to be find appropriate animal or mouse models for something like this, to see if it is having the effect in an animal that we see basically in the test tube.
Yes, I mean, I totally agree. I think one of the things that would be of most interest would be if these molecules actually get across the blood-brain barrier as well. And that, from published data that we've seen around Tilrone, there have been reports that this has activity in stroke. So that would perhaps suggest that it does cross the blood-brain barrier. But it's unclear whether riboxystorin does. So maybe that's one of the first things we have to do. If there are no other questions, I think we can probably draw to a close. One other question. Have you looked at other routes of drug delivery, such as nasal or pulmonary delivery as a dry powder? The easy answer there is no, we haven't, but that's certainly a very good idea. Thank you. It
would certainly be better than intrathecal.
Yes, probably, yeah, less invasive, right? I would just like to thank everyone for joining us today. Our next webinar will be on endocrine disruption by Kimberly Zorn in about a month's time. So please look out for invites for that. And I would just like to thank you all for joining us today, and Patricia for doing the presentation. And thank you again, and take care. Thank you from both of us.